I'm really excited to have the opportunity to tell you today about a project titled Data-Driven Modeling of Distributed Resource Sharing in Honeybee Swarms on the behalf of myself, as well as Golnar Farad and Elizabeth Bradley. So honeybees are a landmark example of eusocial organisms where individuals in the colony have different tasks. And some of these tasks include feeding the young, constructing or cleaning their honeycomb, guarding the entrance to the hive or ventilating it. And all of these cost energy. And one way for these bees to receive that energy is through another caste of foraging bees that would go and forage for food and bring some with them back to the hive inside their social stomachs. And when they're coming back to the hive, they can regurgitate that food and basically provide it to other bees that perhaps didn't have access to food. This behavior is called trophallaxis, and here is a movie showing an example of how this would look like for honeybees. So the bee on the left basically is a donor or a bee that is fed and has food in her social stomach, and it's regurgitating some of this food and sharing it with a more deprived bee. So this behavior is very important for the survival of the colony and has been studied by many different labs. Just to name a few examples, there is the lab from the work from the lab of Ofer Feineman, who can fluorescently label that food and give very precise measurements of how much food is being exchanged and when and where and so on. Another recent example is coming from the lab of Jean Robinson, who can actually track trophallaxis events in an entire honeybee colony, which is very impressive. But what I would say is currently missing is a mechanistic understanding of the local behavioral rules that the bees operate by using only local information, because the colony is very large. They don't have access to the position and states of all the other bees in the colony, so they must be using some kind of local information to make decisions. And this knowledge is also desirable from the perspective of swarm robotics, technological applications, where individual robots can potentially charge each other and also be programmed using local information from the environment. So more specifically, what we are seeking to understand is the answer to this question. So we have a scenario where a bee just fed another bee, and then this bee can basically feed another bee that is nearby or more far away. And the reason to think about this kind of question is because if we can think about the food distribution process as a diffusive process. So we know from just Brownian motion or normal diffusion that the mean square displacement would behave as a linear function of the time times a coefficient, which is a diffusion coefficient. So if a bee basically moves less, it would have a lower diffusion coefficient. So the slope of this curve that we see here on the right side would change. And by moving further away or faster, it can effectively manipulate the value of this diffusion coefficient to either be higher or even perhaps a super diffusion coefficient process where the bee can disseminate different nucleation events where these newly fed bees can now start the whole process again. So what we want to do is come up with the local rules of movement and interaction and understand how they would affect the efficiency of food distribution when thinking about it as a diffusive process. And the approach in my lab combines usually mathematical modeling as well as physical experiments with behavioral experiments with honeybees. We tend to iterate between them, and I would like today to present the results in this iterative way instead of a linear way, because I think it can help us understand something about the physics of behavior of these individuals. So we start with a model, and we're going to basically arrange for agents that move with a correlated random walk, and we can get them to change their effective diffusion coefficient by changing the width of the turning angle distribution that is shown here on the right side. So if this is a very wide distribution, these agents wouldn't move very far. And if it's a very narrow distribution, then the trajectory would be more straight and they would move further. So we have these agents moving in a two-dimensional arena, and then if they encounter another agent within a certain distance, then they would stop moving and start exchanging food. And the way that the food is exchanged is implemented in a simple way. So basically, it's going to be halved between the two individuals. So they will leave that interaction with a similar amount of food in their stomachs. So those are the rules of behavior, and then we just repeat them over time until we get a more or less equal distribution of food in the entire system, which we define looking at the variance of all the amounts of food that each individual has, and making sure that this is below a certain threshold value. So these are the rules. What we see is that I think pretty much in agreement with the intuition that I described a few slides ago. So if you look at the convergence time as a function of the width of the turning angle distribution, we see that when it's very narrow and we have basically more mixing because of that, because individuals would move more, then the convergence time is lower, and vice versa for wider distributions. And just to illustrate this a bit more visually, here are two examples of these simulations. On the left, we have the more ideal case with more mixing, so individuals are moving further in comparison to what we see here on the right side, and you can probably already see visually by eye that the system on the left is going to converge and reach equilibrium faster than the system on the right side. So this is great. Now we have some intuition from this very, very simple model, but we can actually check it with the bees and see with a behavioral assay if this is indeed what the bees are doing, maximizing the mixing. So we worked with colonies of European honeybees, Apis mellifera, divided into two groups. We had bees that had access to food. We call them the fed bees. They're marked with a little pink dot so that we can track their position over time. And then we also had deprived bees that didn't have access to food for about 24 hours. We set the ratio to an extreme where we have very little fed bees to sort of drive the system to that extreme and see how the bees solve that problem. And then we put them in the two-dimensional arena, record it from the top. And here is a typical experiment. This is a bit sped up, and it's probably a little bit hard to see, but we can actually track those pink dots using a traditional computer vision. This is how a typical analysis would look like. So here we are seeing the movement or trajectories of the fed bees shown in red over about 3 seconds, and this is overlaid with the density of all the rest of the bees. And we can follow how this analysis would look like as a function of time. And here is an example of this. And you can probably already see by eye that these longer trajectories turn to be smaller dots. So that means that these bees are not moving very much. And here is a slightly more quantitative description of this. So you can see the average speed of the fed bees as a function of time. We can see that they're consistently reducing their speed. So they're moving less, they're mixing less, and this is really counterintuitive to the intuition that we had previously about the diffusion process, as well as the model that we saw. It turns out that's not the end of the story. And maybe you can even see it here in this slide. So besides the reduction in speed, what we also see is that the reduction in speed is accompanied by clustering. So the bees become more dense, basically. And we can also more quantitatively analyze it using clustering analysis as a function of time. And here is a display of that. So what you see, the y-axis is the cluster size, how many bees are in a cluster as a function of time. And we can see that just before we introduce the fed bees into the system, we have very small clusters of about 10 to 20 bees. And then as soon as we introduce the fed bees into the system, then we get a sharp increase in the cluster size. So they immediately create those clusters. and they range between 30 to 50 bees, and they also increase, so they're sort of coarsening. So there's really an aggregation process that is happening that we haven't considered before for the model. But we can go back to the model and see how this kind of aggregation would affect the convergence time and whether it would improve it or not. So now we go back to the model, we add this one extra rule where if two individual bees are within a certain interaction range that we refer to as R, and we apply this attraction between them, then they would start moving towards each other. And the rest of the simulation is actually exactly the same. So we have the same rules from a few slides ago. I'm not going to repeat them due to time. We repeat it again until we get convergence where the variance of the food distribution is below a certain threshold. And here is how it would look like for this system with the attraction. So on the y-axis, we see again the convergence time. X-axis is the attraction range. And we can see that there is a really large effect on the attraction range, on the convergence time. So it starts when we have very, very short attraction range, doesn't make a big difference. But then the convergent times decreases at this sweet spot at a certain attraction range, and then increases again. And I'm showing here 3 different plots. Each one is for a different value of the turning angle distribution, just to show you that actually the attraction range is making more of an effect on the convergence time in comparison to the turning angle distribution, which was our original intuition. So to show it again a bit more visually, the simulation here on the left has no attraction. The simulation on the right has this short range attraction. And you can probably already see by eye that this simulation would converge faster. Having this result, we are also very curious to continue to quantify this and answer questions like how robust this set of local behavioral rules is. We're currently doing it by changing the density of the bees in our simulations as well as experiments, because this is a scenario that can actually naturally occur in the hive. The bees can exist in many different densities. So we can quantify how the density would affect that dip in the convergence time as a function of the attraction range. Here you see it for 3 different densities. And this is more of an outlook, but where these simulation results are currently guiding a new set of experiments in the lab, where we're also changing the density. So to summarize, I showed you a trophallaxis-inspired agent-based model that is designed and validated using lab experiments on honeybees. Our results show that a more tightly constrained random walk at higher densities with local attractions can increase the efficiency of the food distribution, and we provide this local set of behavioral rules. If you're interested in more details, the paper is currently on the bioRxiv, showing the link over here. And with that, I just want to acknowledge my lab and Golnar, who is the graduate student who led this work, as well as Elizabeth Bradley, who is a co-advisor of Golnar and has given us immense support and feedback on this project. And thank you for your attention. And I'll be happy to take any questions that the conference format would allow me to.